name is Sam Al-Galvani. I'm going to talk about bioprocessing, organism development.
Hi, I'm Matteo Spani. Today I'm going to be taking you through fermentation processes.
Hi, I'm Eileen and I will be talking about product isolation and purification.
I'm Stuart Taylor and I'm going to be talking about product formulation.
Hi, I'm Ahmed Naim and today I'm going to be talking about engineering challenges and biosecurity.
So, what is growth hormone?
Growth hormone is a hormone that stimulates growth in human, animal, or plant cell, and it is a 191 amino acid hormone, single chain polypeptide.
So, where does it come from?
For growth hormones, growth hormone 1 and 2, can be localizing from chromosome 17. Before recombinant DNA technology, the growth hormone gene was extracted from the pituitary glands of corpses. Due to limited supplies of human growth hormone, a synthetic replica of the hormone has been produced. We will discuss the processes by which these are fermented, isolated and purified later on in the video.
So how does it work?
Growth hormone is secreted from the pituitary gland in pulses. Now, these can be triggered by a variety of factors, such as deep sleep, exercise, or the higher concentration of sex hormones in one's body during puberty. There are a number of different means by which growth hormone causes one to grow. It increases transcription and translation, meaning more protein is synthesized. Simultaneously, the growth hormone increases lipolysis, reducing the amount of fat in one's body, and also reduces the amount of glucose taken up by one's liver.
So, what is the uses of this hormone?
It is commonly used for children with growth hormone deficiency or insufficiency, poor growth due to renal failure, Turner syndrome, which means girls with a missing or defective X chromosome, and children born small from gestational age with poor growth past 2 years of age.
So Ahmad, who uses it?
HGH is used for replacement therapy in children and less commonly in adults. Athletes have also been known to use HGH to increase their performance and muscle mass. However, most sports ban its use. Recently, HGH has been promoted to older patients to combat the effects of aging. However, there is no apparent evidence to support this.
Come in. Hello, have a seat. Thank you. Regarding yesterday email, I have researched about bioprocessing organism development, and I found that in 1940s, growth hormone was manufactured by extraction from the pituitary glands of cadavers. As animal growth hormone have no therapeutic value in human, a production of a small amount of this hormone required a lot of pituitary gland, and this of course creating a significant shortage of the hormone. Can you please tell me more about that? Yeah, growth hormone was isolated from the human pituitary gland in 1956, but its biochemical structure was not discovered until 1972. By 1985, pituitary growth hormone was in use for nearly 30 years in the United States and Canada. In the same year, U.S. Food and Drug Administration received reports of 4 young adults in the United States with the fatal who had been treated with growth hormone in the 1960s. On April 1985, distribution of pituitary growth hormone was suspended and use of human pituitary growth hormone was stopped. Identification of the biochemical structure of growth hormone in 1972 became the catalyst for the development of recombinant DNA-derived human growth hormone. For the first time in 1979, it was produced human growth hormone by inserting DNA coding for human growth hormone into a plasmid that was implanted in E. coli bacteria. E. coli and related bacteria possesses the ability to transfer DNA via bacteria conjugation, transduction, or transformation, which allows genetic material to spread horizontally through an existing population. Genetic developed in 1981 the first recombinant human growth hormone by a biosynthetic process. Later, an improved process to develop recombinant human growth hormone was developed called protein secretion technology. So nowadays, and with the development of recombinant human growth hormone, an unlimited commercial sources become available. Sorry, I have no time now to explain it more in details. So see you later.
Fermentation is the bulk growth of microorganisms on a growth medium that takes place in the lack of oxygen. To achieve high cell densities, fed batch processes are used. This process uses inoculant in the system at an optimum concentration of nutrients to maximize growth rate. When creating a growth media for the culture of high cell densities, there are several problems that need to be addressed in order to achieve maximum dry product yield. When exceeding certain concentrations, some nutrients, mainly carbon and nitrogen, can restrict cell division. However, more generally, I've listed other key nutrients that, when present in higher concentrations than specified, will start to inhibit growth. To tackle this problem, high-density cultures are initiated with concentrations below their highest limits shown. There are two types of growth media. Defined growth media. This is where all the chemicals and the concentrations used are known. Also, no yeast, animal, or plant tissue is present. Or complex media. This is where a carbon source such as glucose, water, various salts, amino acids, and nitrogen are used for bacterial growth. In the culture of high cell densities, a defined medium is usually used. This is because the nutrient content is known and therefore can be manipulated to stimulate greater growth patterns. However, occasionally a complex media is used to boost product formation. Acetate is formed as a byproduct when E. coli is grown under anaerobic conditions. However, it can also be formed in aerobic conditions when a glucose medium is used. From previous reading, it is seen that in high concentrations of acetate, the growth rate and biomass yield of E. coli is reduced. The reason for this is that the acetate obstructs the ATP production as a result of the difference in pH contribution to chemiosmosis being reduced. This problem is made worse by the salts that are produced as a result of an acid-base reaction. Techniques such as lowering the growth rate of E. coli by limiting essential nutrients in fed batch cultures have been used to lessen the generation of acetate. In a different approach, dialysis techniques have been used to remove the acetate produced. However, this could be very complicated to scale up and results in large quantities of nutrients being lost. Acetate production can be reduced when glycerol is used in the growth medium. However, glycerol is more expensive than glucose and cultures grow quicker in glucose. By reducing the temperatures of the medium from 37 degrees to 26 to 30 degrees, the nutrient uptake and growth rate are both reduced. The consequence of this is that toxic byproducts and buildup of metabolic heat are reduced. Another simple strategy, such as the exponential feeding rate, allows the control of specific growth rate below the essential value of acetate formation. This seems to be a particularly good technique as it is very simple and does not require any special apparatus.
After the fermentation process, the recombinant growth hormone will undergo a series of distinct unit processes linked together to achieve product isolation and purification. It includes cell harvesting, mechanical cell breakage, centrifugation, ammonium sulfate precipitation, ion exchange chromatography, and gel filtration chromatography. The initial step of any purification procedure involves recovery of the protein from the source from which it is produced. Initial product recovery involves the separation of the whole cell from the fermentation medium by ultrafiltration or centrifugation. Next, we have to disrupt them in order to relieve of the protein of interest. Disruption means breaking open those cells and membrane in order to obtain a desired protein. The cell disruption is often performed by using high-pressure disruption French press or sonication. Following cell disruption, soluble protein are usually separated from cell debris by centrifugation. Here, we spin a sample so that the larger and insoluble components will form a pellet that can be separated from the protein. To proceed, ammonium sulfate is added to the mixture. Ammonium sulfate precipitation is a technique used to quickly remove large amounts of contaminants protein as a first step in many purification schemes.
Ammonium sulfate is highly hydrated such that a concentrated ammonium sulfate solution will be able to reduce the available water considerably. After that, a multi-step purification is required to separate human growth hormone from all the other proteins and to remove nucleic acid and interribosomes. The first step is to be carried out is the ion exchange chromatography. Ion exchange chromatography is based on electrostatic interactions between molecules in the sample and the ion exchange matrix. The ion exchange matrix is either positively charged or negatively charged. Therefore, the separation of the product of interest depends strongly on the pH of the mixture, as the pH will affect the charges of the substrates and thus affecting the binding of the substrate to the ion exchange matrix. For the human growth hormone, the ion exchange chromatography is firstly performed by using positively diethyl aminoethyl cellulose and performed again by using negatively carboxymethylcellulose cellulose. The final stage in the purification process is gel filtration. Gel filtration chromatography, also termed as size exclusion chromatography, it separates protein on the basis of their size. This method is often applied at the end of purification process where the protein of interest is already relatively pure and is present in a small, concentrated volume. The method also results in significant reduction in the concentration of protein in the solution where most of the protein has been absorbed into the gel. And now, we have purified human growth hormone.
Hello, I didn't see you there. Well, we have seen how recombinant DNA technology can be used to produce human growth hormone, known in the pharmaceutical world as somatropin. Once purified, this is ready to be made into a usable product. This process is called formulation. But what does formulation mean, I hear you say? Well, formulation is the process by which somatropin is systematically mixed with a number of other chemical substances to produce a desired product. I like to think of it as following a recipe to make a cake. All of the ingredients must be added in the right order, in the right quantities, and with the right technique, or it simply will not work, and nobody wants that. The aims are to ensure that the formulation renders the growth hormone safe for use, that the human growth hormone does not denature in the process. It must be pharmaceutically viable. For human growth hormone, the formulation can vary, and it will depend on the exact product or pharmaceutical company and the method by which the growth hormone is to be administered. The drug genotropin, produced by Pfizer, comes in a 2-chamber cartridge for injection either by a syringe or a specially designed pen. The dosage of somatropin, the active pharmaceutical ingredient, can vary between 5 mg up to nearly 14 mg. The growth hormone needs to be formulated with the method of administration in mind. Injections of the product need to be at the subcutaneous region, that is just below the bottom layer of the skin. Pioneering research is being undertaken into sustained release options for human growth hormone, with the aim of improving efficacy and reducing the necessary dosage. The method of formulation is also significant. Most of the time the growth hormone is lyophilized, that is to say freeze-dried. However, it may also be milled into very fine particulates with air jets, spray-dried with liquid nitrogen, or even supplied in an aqueous form. A technique called isoelectric precipitation can be used, where the pH of the solution is slowly adjusted using a series of buffer solutions. So, to summarise, the formulation stage is the final putting together of the human growth hormone. It is vital both that the human growth hormone does not denature in the process, and that the drug is safe and does not have any unexpected adverse effects. Cheerio.
There are many challenges in creating human growth hormone that relate to the engineering and safety aspects of its production. Since HGH is recovered from E. coli with undesirable alterations and contaminated with other substances, extensive purification is needed to obtain the desired purity in the HGH. During this process, the hormone can become potentially damaged due to changes in the folding and cross-linkages of the peptides. Molecules may become hooked together and become polymers, causing adverse side effects. Using different cell cultures may remove the need for expensive purification. However, none of these have the same production efficiency as E. coli. HGH is extremely fragile and is completely dependent on retaining its physical structure.
For most HGH, when refrigerated, biological activity can be preserved for up to 2 weeks, after which the product can no longer be used. Products cannot be shipped or stored at room temperatures and must always be refrigerated. However, standard methods of freeze drying can alter its structure. These abnormalities can cause allergic reactions, desensitization to the hormone over time, and autoimmune responses in the body, neutralizing the HGH dosage as well as internally produced HGH. Because of the fragility of HGH, products must be heavily tested and regulated to ensure the HGH is safe to use, as biological activity is not difficult to achieve but does not prove that the product is safe from any alterations or contaminations. A variant of HGH is known as HGH20K and is believed to have less diabetogenic effects on people, which is a side effect that occurs with prolonged use of HGH22K, the most commonly used human growth hormone. However, it is much more difficult to industrially produce HGH20K, as it cannot be produced efficiently by the intracellular method using E. coli. As for the safety aspect, E. coli is generally harmless and is found inside human intestines. However, with any bacterial culture, there is always a chance of mutation or pathogenic contamination. Some strains of E. coli can cause several diseases, so caution must always be exercised. Lab coats, safety goggles, and gloves are extremely important when handling or near to the E. coli, and food and beverages should not be allowed to be in the vicinity of the bacteria culture. Gloves should be disposed of in biohazard labeled bags, and hands should be washed with disinfectant.